Okay, so our first talk today is from Dr. Kristen Brown, who is in Sharon Davis's group. And her talk is about a multi-omics approach to understanding diabetes risk in African Americans.
Thank you so much for the introduction. I'd also like to thank the Institute for the opportunity to present today, as well as to my branch, the Social and Behavioral Research branch, for the nomination to do so. So as stated, the title of my talk today is Cracking the Zip and Genetic Codes, a multi-omics approach to understanding diabetes risk in African Americans. So in the way that we traditionally approach science, we do it in the silos of our own disciplines. So as geneticists, we have our GWASs and our polygenic risk scores and our sequencing, and we do our genetic work and we write papers, and then we pretty much write them for other geneticists to read and try to increase our understanding. And then in a different silo, we have the nutritionists, and the nutritionists look at dietary patterns and different macronutrients and try to figure out how to use food to optimize health. Then in a different building, we put our epidemiologists. In our epidemiologists, we have our surveys and our fancy statistics and mediation analyses, and we try to figure out disease trends and disease transmission. And then in another building, we throw our pharmacologists who are trying to figure out new drugs and new therapeutics and correct dosages. But in this era of precision medicine and multi-omics research, we're challenged as scientists to build bridges across our different fields as an optimal way to approach health and disease. And this is the angle that I'm coming from in this presentation today. So I asked the big picture question: if we bridge knowledge from the social epidemiologic and genetic literatures, can we do better at predicting diabetes than either field could alone? So when we look at the social epidemiologic side, we know that the neighborhood environment is important for risk of diabetes. So things like access to healthy food, your opportunities to engage in physical activity, as well as the social environment, questions like, do your neighbors get along, or is there a lot of crime in your neighborhood? We know that those things are associated with diabetes. Interestingly, this holds even after accounting for individual level behaviors. So living in a neighborhood where there's opportunities for physical activity is good for your health, even if you're not intentional about engaging in those physical activities yourself. And then when we look on the genetic side, we know that we've identified gene variants associated with type 2 diabetes from our GWAS studies. So these variants have been combined into polygenic risk scores, which we then have been able to predict incident diabetes in various sample sizes. So for this project, I asked the question: if we put together what we know about a person's neighborhood, combine it with their polygenic risk, can we predict fasting blood glucose better than either factor could have alone? So for this project, I used data from the gene forecast study, or the genomics, environmental factors, and the social determinants of cardiovascular disease in African Americans. It's a study of US-born African Americans aged 21 to 65 at baseline, who are currently living in the metropolitan Washington, DC area. The participants complete an in-person visit to the NIH Clinical Center, where we're able to do surveys as well as do lab work. It's an ongoing study, and currently we have 298 participants for whom we have both the neighborhood and genetic data available for this analysis. Looked at two main exposures, the first exposure being neighborhood level factors, looked at 3 neighborhood level factors: socioeconomic status, physical environment, and social environment. So neighborhood socioeconomic status was measured as a weighted sum of 7 census tract level variables. So those are things like median household income of the neighborhood, percent poverty, percent unemployed, for example. The physical environment we did via survey, and the survey had a Likert scale responses. So those are the surveys that you fill out that say strongly agree, agree, neutral, disagree, strongly disagree. An example question may have been, there's a lot of opportunities to buy fruits and vegetables in my neighborhood, or I see a lot of people exercising in my neighborhood. And for the social environment, it was also measured via survey with the Likert scale responses. And the example may have been, my neighbors get along. Do you strongly agree, agree, neutral, disagree, strongly disagree? Our second main exposure was the polygenic risk score. So we used a polygenic risk score that was based on 65 SNPs identified from the Diagram Consortium. So the Diagram Consortium is a very large consortium that studies diabetes in Europe. It has a very large sample size of approximately 150,000 people in it. And we use this risk score because it was previously found to associate with incident diabetes in African Americans in the NHGRI funded CARDIA study. And for our main outcome, we looked at fasting blood glucose. Fasting blood glucose we measured from a blood sample collected from participants during their NIH Clinical Center visit. Glucose was measured using the acute care panel and measured in milligrams per deciliter. So to address our big question, we broke it up into 3 smaller questions. So the first question simply being, are neighborhood factors associated with fasting blood glucose in gene forecast? And what we found was that neighborhood socioeconomic status and physical environment were associated with fasting blood glucose, but we did not find a significant association for the social environment. Secondly, is a polygenic risk score from the Diagram Consortium associated with fasting blood glucose in gene forecast? Interestingly, we found that there was not a significant association between the polygenic risk score and fasting blood glucose in our study sample. And third, does including the neighborhood factors and the polygenic risk score in the same model predict fasting blood glucose better than either could alone? Well, as you would expect from question 2, where the genetic risk score was not associated with fasting glucose, we also found that including the genetic risk score did not improve upon the neighborhood-only models. So what does this all mean? It really highlights the importance of healthy neighborhoods. So we found that neighborhood factors are associated with fasting blood glucose, even after accounting for individual level factors. Such findings are consistent with the epidemiologic literature about the associations between neighborhood and diabetes. And it highlights that intervention studies should prioritize developing and sustaining healthy communities. When we look at the genetic side, we found that the polygenic risk score was not significantly associated with fasting blood glucose in the gene forecast study sample, and that more work needs to be done before polygenic risk score should be universally used in clinical practice for African Americans. So this finding that the polygenic risk score wasn't associated with fasting blood glucose in our study sample was not totally unsurprising. So here I highlight a figure showing the intercontinental portability of polygenic risk scores developed in European samples. And as you can see, the prediction accuracy of polygenic risk scores developed in European samples is reduced across continents with the lowest prediction accuracy among Africans, which is reflected in the study that we present today. And if we take a step back and think about why that may be, well, our polygenic risk scores come from our genome-wide association studies. So here I highlight a graph from the GWAS Diversity Monitor. And as you can see, an overwhelming majority of participants in GWAS studies have been European or European descent individuals. If we then look by studies themselves, we can see that over time, there's starting to be a greater percentage of GWASs in non-European samples. However, these studies have smaller sample sizes, so the issue of equitable representation remains. And it really highlights that there's an important need to diversify our genetic studies to gain equitable access to scientific advancements. Future directions. So I think the immediate future direction that we can do is to use the diabetes-associated SNPs identified in African descent, or multi-ethnic genome-wide association studies. We're starting to have an increase in GWAS studies that have diverse samples, including work being done in our own institute. Secondly, is to evaluate whether the neighborhood environment affects genomic regulation in this new area known as human social genomics. And lastly, is to identify which aspects of the neighborhood are most important to health outcomes. With that, I'd like to acknowledge all of my collaborators as well as the study participants. And I thank you for your time and am welcome to taking any questions.
Excellent talk. So we have some questions already coming in. So I'll just jump right in. So one person asks, do you think it would be important to look at where people have lived in the past rather than, or in addition to, where they're currently living? And then also ask, if that is important, then how do we deal with that sort of privacy concerns when you are asking about people's entire history?
Great question. So yes, I do think that it is important. And there is literature that talks about childhood environment as well as your current environment, because to some extent, your childhood environment is shaping, the trajectory that you go on in adulthood. So I think that having this life course perspective is actually a better picture than just looking at the slice of time in which we're doing our study. In terms of privacy, I think that it's an exposure. It's an exposure in the same way that we handle all of our exposures in science, that understanding the trajectory of a person's life is also important to health as well. So that's a great question. And yes, I do think we should, at best as we can, look at the longevity, as much information as we can actually have.
Okay, and I think we have time for one more question. This is actually two separate people asked. Did you look at HbA1c?
No, I didn't. We actually had that available in our study, so I do have the capacity to do so. But for this study, we just use fasting blood glucose.
Okay, excellent. All right, and there were several other questions, and I think we can answer those in the text, and we'll move on to the next speaker.